Hello everyone, thank you so much for joining us for today's webinar on understanding patient randomization and the role of interactive response technology, or IRT, in clinical trials. My name is Melanie Ciotti. I'm our marketing strategist here at SDC, and I'll be moderating today's webinar. We have two great speakers for you today, but before I introduce them, I wanted to go over a few webinar housekeeping tips. First, you have two audio options for listening in on today's webinar. You can either use your computer speakers or you can call in by phone. If you have a poor or a slow connection, or if you hear any interference on the line, I would recommend switching your audio source to try to troubleshoot that. Also, this is a live webinar, which we are recording live. So if you need to step away for any reason, or if you have technical issues, we will go ahead and send out the link to the recording so that you can watch this at a later date. You may have noticed that the audience is on mute. If you have any questions throughout the presentation, we do encourage you to ask them using the questions section in the webinar portal, as you can see indicated on your screen. We should have about 10 to 15 minutes available at the end of the presentation for a Q&A session. So please go ahead and submit your questions throughout, and we'll go back through all of them at the end. If we're not able to answer all of the questions, we'll also do our best to follow up with those individuals via email. It's my pleasure to introduce our speakers for today's webinar. First up is Susan Potts, Senior Principal Biostatistician. Susan has over 25 years of experience as a clinical trials biostatistician, and she's been a member of the SDC biostatistics team since 2013. She's supported over a dozen FDA product approvals in her career, as well as several global product approvals to international regulatory authorities. Her experience spans a wide range of therapeutic areas, just a few of which we are listed here. Prior to joining SDC, Susan spent most of her career on the sponsor side at both small biotech and larger pharmaceutical companies, such as Alcon Laboratories, where she worked for many years. Susan is a subject matter expert on patient randomization, and she'll be covering that topic for us today. Jen Ohme is SDC's resident expert in interactive response technology, or IRT. As IRT project manager, Jen oversees and coordinates all IRT activities in support of clinical research studies. This includes everything from IRT system selection and validation to site initiation and setup, and continuing on through study conduct, maintenance, and closeout. She has over a decade of experience in the healthcare technology arena and specializes in e-clinical solutions for complex clinical trials. She has supported clinical trials across a diverse range of therapeutic areas for both drugs and medical devices, including but not limited to those therapeutic areas listed on the screen. As an expert in this area, Jen will be covering the IRT portion of today's webinar. Here's our agenda for today. Susan will begin with the importance of randomization in clinical trials, as she explains patient randomization and blinding or masking a bit further. She'll share some examples of complex randomizations and treatment assignment schedules. Then Jen will take over and explain about what IRT is and the benefits of using an IRT system for patient randomization and inventory management. Again, we should have some time left over at the end for questions and answers. So please go ahead and submit them as they come up using the chat portal or the questions portal on your webinar pop-up. Thank you again for tuning in to today's webinar by SDC, and it's my pleasure to introduce you to Susan Potts about patient randomization.
Hello. Thank you for joining us today. I'm going to cover the importance of randomization in clinical trials as my first topic. So my language arts teachers would be very, very pleased to see that I've incorporated the 5 W's into my presentation for randomization. So they'd be proud to see an English topic being included in a scientific presentation. So what I'll cover are the 5 W's of randomization, which are seen here. Who benefits from using randomization? What do randomization and blinding accomplish? When is maintaining blinding difficult or unnecessary? Where can randomization break down? And why do some randomizations require an IRT? Who benefits from using randomization? First, and most importantly, patients benefit from randomization. As an example, if a clinical trial is designed to assign patients to active study drug or placebo with the same probability, each patient has a 50% chance of receiving a potentially therapeutic treatment for their disease. These probabilities are so important in a randomized clinical trial that they are carefully detailed in the informed consent form so patients are apprised of the potential to receive a beneficial treatment. Sponsors, companies looking to provide efficacious and safe treatments for diseases, can assess a new treatment for a disease in a fair and unbiased setting. Randomization also has the propensity to increase patient recruitment because patients are educated with regard to the probability they will receive the new experimental treatment compared to receiving standard of care or placebo. Data analysts, randomized clinical trials yield an estimate of the effect of a treatment that is unbiased and consistent. It is critical that all data analysts remain blinded to the randomized treatment codes until after database lock has occurred and all analysis populations have been identified to prevent any analytical bias that could occur. And lastly, regulatory agencies. Regulatory agencies require that randomized clinical trials be conducted to minimize confounding and bias in the data that are collected. These are critical factors in determining if the new drug is truly efficacious and safe. What do randomization and blinding accomplish? Randomization is a powerful method to prevent confounding. Confounding is the bias that occurs when one group of patients have features, known or unknown, that affect the relationship between the intervention and the outcome of interest. If a large number of patients are allocated to treatment groups by a random method that gives an equal chance of being in any treatment group, then it is likely that the groups will have similar distributions of age, sex, behaviors, and virtually all other known and yet known possible confounding factors. As sample sizes decrease, the likelihood of confounding increases simply due to chance. Successful randomization is dependent on allocation concealment, which refers to preventing the next treatment assignment in the clinical trial from being known. If the next group to which a patient will be allocated is known, the randomized clinical trial has the potential to become a non-randomized clinical trial. Masking. I must tell you that you will hear Jen and I both interchangeably use the word masking and blinding. I've worked in the ophthalmology field for 25 years, so we don't typically like to blind our patients. Randomization in clinical trials involves more than generation of a random sequence by which to assign patients to treatment groups. For randomization to be successfully implemented, the randomization sequence must be adequately blinded so that investigators, study staff, and patients are not aware of the upcoming treatment assignment. The absence of adequate blinding can lead to selection bias. Bias can also be imparted in clinical outcomes if investigators are aware of treatment assignment. Patients can change their behavior or self-assessment of key study endpoints, such as patient-reported outcome questionnaires. Committees charged with determining whether patients have achieved a study endpoint may be biased in their assessment if they are unmasked due to preconceived opinions of expected outcomes. When is maintaining blinding difficult or unnecessary? Randomized clinical trials. Post-randomized blinding can be difficult or impossible in certain cases, but it is imperative that allocation concealment be maintained. If you recall from a couple slides ago, allocation concealment refers to preventing the next treatment assignment in the clinical trial from being known. A knee surgery study is a good example of this type of randomized clinical trial. Due to the surgical incision in patients who are randomized to have ACL surgery, post-randomization evaluations are impossible to conduct in a blinded fashion due to the incision. But in this same study, each new patient who is enrolled has no idea of the treatment they will be allocated to, nor do the investigators or study staff. Although there may be some bias in the outcome assessment, the allocation concealment should help to minimize that as much as possible. Single treatment clinical trials are often required for registration of a new drug. There are certain types of these clinical trials that do not require randomization or blinding. An example of this type of clinical trial is a long-term safety study that does not include a comparator. These are required by regulatory authorities to support the long-term chronic use of the molecule being studied. Given this objective, all patients who are enrolled into these types of trials are assigned to the investigative treatment under study to gain as much safety information as possible. Another example is a compassionate use study. Often, sponsors want to ensure that all patients in their phase 3 clinical trials continue to receive, or if on the placebo side, are given the opportunity to receive an effective treatment for their disease. Patients from these phase 3 clinical trials are enrolled into a compassionate use study and all receive open-label active study drugs. Where can randomization break down? Incorrect randomization number assigned to a patient is an example. An incorrect randomization number can inadvertently be assigned to a patient at the beginning of the randomized clinical trial. As an example, the first patient should have been given randomization number 1001, but the study site staff mistakenly gave them the treatment associated with randomization number 1002. Often this mistake is not discovered until the next patient is enrolled into the study. The site staff assigns the second patient randomization number 1002, but when they enter the clinical supplies room to retrieve the treatment number 1002, they discover that 1002 is missing and 1001 is still on the shelf. At that point, the clinical team and statistician are notified of the mistake, and a flurry of activity ensues to derive a solution. Should the second patient simply be given randomization number 1001? Should the first patient be discontinued? If so, what randomization number is given to the second patient because 1002 is not available? As you can see, this is a difficult scenario to deal with, regardless of the decisions that are reached. These types of mistakes also have analytical consequences. Efficacy analyses must be performed for both scenarios. In other words, we must analyze the efficacy data using the treatment the patient was supposed to have received, as well as using the treatment the patient did receive. Safety analyses must be performed using the treatment the patient did receive to ensure that any safety signals are attributed to the treatment that was given. A second example is when an incorrect treatment kit is dispensed. When a new kit is to be dispensed at each visit, the likelihood of dispensation errors increases. As an example, if a patient is given a randomization number that is associated with the active study drug, that patient's kit numbers should all be associated with active study drug. As shown on this slide, you can see that the patient received active study drug kits at the first 2 visits, but was erroneously dispensed a placebo kit at the 3rd visit. As in the above scenario, these errors may not be discovered until the next patient comes into the site to receive his or her kit, and the study staff discover that the correct kit is not on the shelf, and a kit associated with the previous patient is still sitting on the shelf. Again, you have the same flurry of activity that occurred in the first scenario. The analytical aspects are even more complex than the previous scenario. Do you perform the efficacy analyses using the planned treatment for all visits? Do you perform the efficacy analyses using the actual treatment given at each visit? For safety analyses, you have the same issues to deal with. As an example, if a patient had an adverse event that occurred only during the time she or he was on the incorrect treatment, do you attribute that adverse event to the treatment the patient was given at the time the AE occurred? Why do some randomizations require an IRT? First, I'll go into some simple manual non-automated scenarios. An example of a simple randomization that can be manual or non-automated occurs when a patient is randomized and receives a single treatment for the duration of the randomized clinical trial. Each site would have a list of randomization numbers that are assigned to patients in a sequential fashion. A randomized clinical trial that does not require inventory management could occur if the study drug is abundant and sites receive enough drugs up front for all of their randomization numbers. In this type of clinical trial, there can be a large amount of study drug that is wasted. Alternatively, if study drug is very expensive or difficult to manufacture, inventory management could be required to trigger shipments to sites when their supply drops below some critical value. Prior to IRT, these types of triggers were managed by the investigative site and involved direct communication from site to whomever was managing study drug inventory. Automated unblinding may not be desired if the study drug has a scratch-off label or unblinding envelopes are stored at each site. Alternatively, an IRT can be used to unblind patients by authorized users entering pertinent information into the IRT system. IRT unblinding generates an audit trail in real time, whereas non-automated unblinding is documented in the CRF and is not usually in real time due to data entry delays at sites. Complex automated randomizations that I'll cover on the next 3 slides will go into detail regarding 3 examples that require an automated system. The examples that will be presented involve dispensation of multiple kits throughout the duration of the randomized clinical trial, complex stratified randomizations that become burdensome without an IRT, and finally, dynamic randomizations that simply cannot be handled without an automated system. I'm going to compare and contrast the 2 examples shown on the slide. A simple randomization with multiple kits can be handled by using the randomization number to label the kit. In this situation, the kits employ a randomization schema where a different letter is appended to the end of the randomization number for the kit, and each of the kits is dispensed at the appropriate visit. For example, the kits could be labeled as displayed on this slide: 2001A, 2001B, and 2001C. And 2001A would be dispensed at the first dispensation visit, 2001B dispensed at the second dispensation visit, and 2001C dispensed at the third dispensation visit. This type of kit labeling can lead to wasted study drug if the patient discontinues from the study because his or her kits cannot be dispensed to another patient. An example of a complex randomization with multiple kit dispensations would require an IRT if the kit numbers are scrambled and each site receives kits that are not numbered sequentially. As an example, site number 1 might receive kit 10017, 12947, 18999, and 19721 in their first shipment. The IRT system uses the randomization number, which is tied to a particular treatment type, to dispense study drug kits that are of the same treatment type as the randomization number. This would be impossible in this situation to handle without an IRT unless each site had an unblinded person to dispense kits of the appropriate treatment type to each patient. Stratified randomization. Stratification may involve clinical features that can influence outcome risk. As an example, a testosterone study may require differing treatment based on sex and age group. It is possible that older men may need more testosterone than younger men, and older and younger females may need less than either group of men and may also differ based on age. By stratifying on these 2 categories, you divide the clinical trial patients into 4 homogeneous subgroups. Randomized treatment assignments would occur within each of these 4 strata to maintain balance between treatment groups within each stratum. In the example on the screen, there are 50 females less than 65 years of age in the active treatment group and 48 females less than 65 years of age in the placebo treatment group. You'll see those numbers in the top left green box under each treatment shown on the screen. Although the numbers of patients are not exactly equal in each treatment group within this stratum, in other words, 50 women versus 48 women, this is considered well balanced between the two treatment groups within this stratum. You will note that the other three strata are also well balanced between the two treatment groups. Additionally, the two treatment groups are also well balanced across the study, as you have a total of 201 patients in the active treatment group and a total of 199 patients in the placebo treatment group. If an IRT is not used in this example, each site will need 4 separate randomization lists, one for each stratum. As you increase the number of stratification categories, you must increase the number of randomization lists accordingly. To illustrate, if you have 3 stratification categories with 2 factors in each category, you would need 2 times 2 times 2 equal 8 randomization lists at each site, one for each of the 8 strata. You can see this has the potential to increase human error at sites simply due to the sheer number of randomization lists being used. If an IRT is employed, the site would simply enter in the responses for each of the 3 stratification categories, and the system would allocate treatment based on these entries, thus reducing the propensity for error. Increasing the number of stratification categories or increased levels within strata lead to fewer patients per stratum, which can lead to an overall study imbalance. As study sample sizes decrease, this becomes even more of a problem as some of the strata may have few or no patients, which defeats the purpose of stratification. Dynamic randomization. Minimization algorithms can effectively manage the following: multiple stratification factors such as site, sex, age group, etc. Very few patients per stratum are expected. A minimization algorithm changes the probability of treatment assignment based on the treatment assignments that have already been completed within the system to keep the treatment groups balanced within each of the stratum. This would be impossible to handle without an IRT because the IRT calculates and modifies treatment assignment probabilities based on values that are determined during IRT specification development, which Jen will cover in her topic portion. Minimization algorithms balance the potential imbalance within a stratum as well as across the study. In other words, they reduce imbalances across variables between treatment groups. Adaptive clinical trials are just what they sound like. During the study, decisions are reached, often based on interim analyses of the data or based on recommendations of data safety monitoring committees that necessitate modification. In other words, adaptation of the trial during study conduct. An example study would be one in which 3 concentrations of a compound are being evaluated. During interim analysis and data review, it is discovered that the lowest concentration is not effective. The IRT has the capability of essentially flipping a pre-programmed switch to stop assigning patients to this lowest concentration. The IRT also has the capability to update treatment assignment probabilities from 33% chance to receive one of the 3 original concentrations to a 50% chance to receive one of the 2 remaining concentrations being studied. In conclusion, randomization is very important in clinical trials because it helps ensure that the clinical trial results are not confounded by unknown or known patient attributes such as age, sex, or disease status. Sometimes randomization can be simple, as in the example I shared where each patient was allocated a randomization number and was dispensed a single kit for the duration of the study. But other times, as I discussed in the last few slides, randomization can be quite complex, and an IRT system can play a large part in minimizing risk, ensuring your study runs smoothly and efficiently and with less waste of study drug. At this point, it's my pleasure to introduce Jen Ohme, SDC's IRT expert, to explain a bit more about what IRT is and how it can be useful in supporting randomization and other important aspects of your clinical trial.
Thanks, Susan. As Susan and Melanie mentioned, I'll be providing an IRT overview, as well as discussing the benefits of utilizing and steps for implementing an IRT for your clinical trial. Interactive Response Technology, or IRT, provides various modules or functionality for managing subject interactions and inventory supplies throughout the trial lifecycle. IRT is a standard industry term used when discussing automated randomization and/or trial supply management. An IRT may include IWR or IWRS, which stands for Interactive Web Response System. IWR is website interfacing only for the end user. The end user logs into the website, providing answers to pre-programmed questions, and receives the randomization and/or study inventory assignment. Emails and/or fax confirmations of the data recorded and decisions may also be included. An IRT may include IVR or IVRS, which stands for Interactive Voice Response System. IVR provides similar functionality to an IWR, but is telephone interfacing only for the end user. IXR, or IXRS, is the acronym for an IRT system providing both IVR and IWR functionality, so again, containing both phone and website end user interfaces. The popularity of IVR systems has decreased significantly due to laptops, tablets, etc., with internet connectivity becoming more commonly available in the clinical setting. However, there are studies where having IVR or the phone interface in addition to the IWR may be advantageous, such as when randomization is critical time sensitive or depending on geographical study regions. IRT supports clinical trials from simple to complex by automating the following functionality. In addition to activating and deactivating sites, admin users can set, update, and monitor screening and enrollment caps, as well as set, assign, and update inventory supply limits, all at the site level. Study admin users can open and close cohorts, set and update cohort limits, and manage cohort patient replacement. Site end users can view cohort availability and subject assignments and receive notification of cohort updates during study conduct in real time. Again, IRT replaces manual randomization, so envelopes or scratch-off cards, providing automated randomization. Randomization caps by specified factors, which may include site, strata, country, can also be set and monitored and updated in real time. Screening, rescreening, and screen failures, including screen failure date and primary reason, can be captured and reported on. The inventory lifecycle from depot through subject assignment and return or destruction can be managed and tracked within the IRT. Blinded or unblinded drug dispensing based on treatment assignment, visit, dose titrations, or other protocol factors can be accomplished. And finally, 24/7, 365 by user role, emergency unblinding functionality may be included, including project team notifications. There are numerous benefits to utilizing an IRT for patient randomization and/or trial supply management, including the following. The IRT assigns patients to treatment arms while maintaining blinding and permits complex stratification and randomization designs, keeping balance between treatment arms. Again, as Susan mentioned, an IRT is required for adaptive and dynamic randomization, as previous randomization assignments are used to reduce imbalances across variables and treatment assignments. Trial supply management functionality provides automated initial and resupply shipments to sites, and these can be based on specified factors, which may include site activation, subject screening or enrollment, upcoming subject scheduled visit windows, which is known as predictive resupply, site inventory thresholds or buffers, and inventory expiry dates. These values can all be set and updated as needed during study conduct. Having this flexibility, awareness, and automation assists in using just-in-time methods for inventory shipments and packaging. Inventory isn't sitting at a site when it isn't needed, and at the same time, a site isn't without available inventory when needed. Notifications can be implemented, providing transparency and notice to the broader project team of critical project events, allowing for immediate response. Some of the common IRT notifications include randomization, inventory assignment and dispensation, treatment discontinuation, and emergency unblinding. Reports of critical trial data metrics allow the project team to see the data immediately and to productively act on it. Adding sites, subjects, and cohorts, as well as removing treatment arms during study conduct, can all be accomplished when utilizing an IRT. As Susan touched on, there can be human error when randomizing and/or assigning inventory, especially for studies with complex randomization and/or multiple treatment assignment visits. The IRT reduces this error through automation, including edit checks and workflows, and simplifies subject randomization and/or inventory assignment for participating sites. A few examples would be a subject is not able to be randomized in the IRT until the stratification data is entered, or a subject is not assigned inventory until randomized. And again, a subject is auto-assigned the inventory at subsequent visits based on their treatment assignment and/or other programmed protocol factors. And finally, role-based security, or user roles with specific rights and views, assist to maintain the blind. Randomization and inventory assignment occurs while maintaining the blind, and only specified user roles can unblind within the database when necessary, and as Susan mentioned, with the actions tracked completely within the audit trail. There are several factors to consider when determining if an IRT will benefit a clinical trial and which IRT solution will best meet the trial requirements. As was mentioned previously, a smaller study with one or a few sites with simple randomization and a single treatment visit can be tightly managed with non-automated randomization and may not have the technology budget for an IRT. However, a more complex study with stratified randomization and multiple treatment visits will benefit from utilizing an IRT. Additionally, multiple site regions may require an IRT for 24/7, 365 randomization, if not stratified by site, emergency unmasking or unblinding, and support. Similarly, drug approval by country or managing multiple depots may require use of an IRT to maintain the visibility into the inventory stock and location and country approval status. Finally, for studies with limited or costly products, an IRT may be utilized to provide the increased trial supply management transparency, flexibility, and control needed to reduce inventory waste. When utilizing an IRT for your clinical trial, not all IRT platforms provide the same functionality. It is important to review and understand the IRT functionality within the selected platform to ensure it meets your project requirements. This is especially true when utilizing a pre-validated IRT SaaS or software as a service solution. Two of the platforms we utilize at SDC, iMedNet and Metadata Balance, are pre-validated products offering project-specific configurations. Benefits of a pre-validated product may be shortened build timelines, reduced validation timelines, as only the project-specific configurations may be tested, and reduce overall IRT technology costs. However, if the product configurations allowed do not meet your project requirements, custom coding cannot be implemented in these platforms. Therefore, for more unique project requirements, there are also hybrid IRT platforms such as Endpoint and Ohme, which include both configurations and custom coding. Understanding the end user interface and navigation is also a consideration. One of the IRT platforms we utilize at SDC, iMedNet, is an integrated platform with the EDC. Having one integrated database for EDC and IRT allows for single sign-on for end users, as well as no duplicate data entry. This results in reduced data entry errors and training support time. There are also IRT products and platforms that interface with other trial systems, such as EDC and the CTMS. Interfacing systems, such as metadata balance, allow data to be pushed and pulled between the platforms, again, allowing for reduced duplicate data entry and ease of use for the end user. Interfacing systems may require additional work upfront in the requirements, build, and validation phases of the project, and may still require users to access multiple platforms depending on the action taken. Finally, there can be disparate or separate IRT platforms where the IRT and EDC or other project systems do not talk or interact with each other. Disparate systems may give you more flexibility with requirements and data collection while requiring some additional work and time for the project team and end users. Not all IRT companies provide the same scope and support levels. It is important to understand who is responsible for IRT support, including response and resolution times, to confirm it meets the project team's needs and expectations. When implementing an IRT, step one should always be to identify and assign all project stakeholders to the project team. If not already done, next select the IRT system in looking at your project-specific requirements, budget, and timelines. Your CRO partner may have recommendations for an IRT. If you are selecting a new IRT, we recommend a demo and an audit at a minimum. Once you have selected the IRT, you will work with your project team to draft the IRT user requirements specification, or IRT URS, defining the project-specific IRT functionality required, including the following: the randomization design, the trial supply management requirements, data collection fields, reports, access permissions, and notifications. The level of IRT requirements or documentation can vary depending on the IRT system utilized. After the IRT URS is drafted or approved, the system build begins. And again, this can include configurations and or custom programming depending on the IRT platform selected and project-specific requirements. A lot of companies are moving to a more agile build or validation process, so typically a preview of the database or something similar will be included during build. Having a preview allows for a more interactive review of the resulting functionality and is a good opportunity to make any necessary updates to the specifications before the validation and testing begins. Again, build times can vary widely depending on the IRT platform utilized. After the build is complete, internal validation is executed, followed by external UAT or user acceptance testing. Validation is vital because of the impact on patient randomization, inventory dispensation, and blinding requirements. Client UAT is always offered to sponsors, and even when it is not required, it should be utilized. UAT is an opportunity for the sponsor and all project stakeholders to test and review the system to make sure it meets their requirements. Although we don't require UAT at SDC, we do stress the importance of UAT and perform a UAT handoff meeting with the team to ensure everyone is comfortable navigating through the system to successfully perform UAT and understands the functionality and any high potential risk areas to focus on. After UAT is complete, the system will be released to production and sites and trained users activated. Once the database is live, there is ongoing IRT support and training and change control management. Overseeing or receiving data from the help desk, looking at any support request trends, retrainings needed, or items to be included in the newsletters. Managing any project releases, as well as product releases depending on the platform that may occur during study conduct. And again, this will include keeping the system validated and keeping the sites informed of any impacts to the database because of the releases, including any system downtime, any changes to the functionality, and any retraining needed. So what not to do when implementing an IRT system? The first is to not implement an overly complex system. The goal should be to design an easy-to-use, straightforward system that meets your requirements and protocol. You also do not want to implement a completely inflexible system. You want to balance EditCheck programming and system workflow with the need to accommodate human error. There may be out-of-visit windows, misrandomizations, and misdispensations. You want the system to assist in reducing these errors, but they may still occur, and the IRT needs to be able to accurately collect the data. At SDC, we utilize an IRT plan, which includes how misrandomizations and misdispensations will be tracked, handled, and communicated during study conduct, and have built-in edit checks and notifications to the IRT PM if and when they occur. You also want to make sure you are obtaining input from all stakeholders up front. When the initial site shipment is triggered to the depot, this should not be the first time the depot is seeing a notification from or interacting with the IRT system. Again, step one should be to identify all project stakeholders so they can be included in the IRT URS, previews, and UAT. And finally, you want to ensure all members of the trial team are properly trained, allowing them to easily navigate through the system and leverage the power of technology and reduce data entry errors during study conduct. In summary, randomization is critical. It avoids confounding and maintains statistical validity of the clinical trial results. As we've discussed today, it can be manual or non-automated for simple randomization or automated via an IRT system for more complex. And it helps ensure safe and effective treatments are approved and made available to patients. IRT, or interactive response technology, can provide IVR, IWR, or both as the end user interfaces. And again, it automates randomization and/or trial supply management and can be critical when executing complex treatment schedules to reduce that human error. Thank you.
Thank you so much, Jen, and thank you, Susan, for your presentation. We do have quite a few questions that have come in. If you have any questions that you've been waiting for, please go ahead and submit them through that questions box in the webinar portal. We'll go through some of the questions I have now. It looks like we have about 13 minutes left for the presentation. So we'll get through as many questions as we can. And for those who have questions that we're not able to address, we'll try to follow up via email in the next week or so. Here's one for you, Susan. It was pretty early on, around slide 9. It says, can you give an example of unknown subject attributes?
Sure. Most of the examples I gave were for known subject attributes. We know if someone is a male or female, their age, and so on. But within those groups, there might also be unknown attributes, like a biomarker or some underlying medical condition that is not being collected in the study data. As an example, you may have an opioid clinical trial, and they've actually done research to note that most people have a biomarker, and if it's positive or negative, it can predict the outcome of whether they're going to be responsive to an opioid or not. So randomization helps minimize the potential for these underlying or unknown factors from introducing bias in the data, such as that biomarker not being collected.
Great, thank you. Here's another question. This one's about inventory management. Can you explain more about how inventory management works?
Sure, I'll take this one. So for inventory management, say you have multiple sites for your study, so sites A, B, and C, and at the study start, you're expecting each of these sites to enroll 100 patients over the next 1 to 2 years. Rather than shipping 100 treatment kits to each of these 3 sites and risking loss, damage, etc., you can set up within an IRT automated resupply shipment triggers. So each site at activation could be shipped 10 kits, and then as they're enrolling, once their inventory is down to a set threshold, say 5, you could then ship them another 10 kits. And again, this process of resupply automation would keep going through study enrollment. This way, if you have a site, one of the sites is not as actively enrolling, say they only enroll 30 subjects, you don't have 70 kits sitting at that site. And it gives you flexibility if the other sites then are enrolling additional patients to move those kits or ship those kits to those sites, or even if needed, open up another site and ship kits to them.
Great. Here's one back about randomization for you, Susan. How does randomization work if the statistician is blinded?
That's a great question. As an example, for a double-blind study, the project statistician would be blinded and not able to see whether a patient is on the active treatment or placebo. There's typically an independent, unblinded statistician who creates the initial randomization and then creates the final randomization once that dummy randomization is approved by all stakeholders, and they manage any interim analyses that might be conducted. So this individual is often a member of data safety monitoring committees or data monitoring committees. The project statistician is still able to run all the SAS programs and prepare the analyses using dummy data, the initial randomization that was created for approval. And then at the end of the study, once the database is fully locked, the unblinded statistician will send the official randomization listing so that analysis can be performed with the actual treatments that were assigned to subjects.
Great. Let's go back to the IRT section. Here's a question about how much does IRT cost on average? Jen, do you have a range you might be able to share with the audience?
Sure. So it definitely depends. It depends on the IRT platform, the functionality that you're implementing, and then the duration of the project. But we've done smaller studies, maybe randomization only, that can be under 50,000. And then, as you can imagine, for the larger phase 3 or international studies with multiple depots and complex treatment schedules, it can be over, I think, 500,000 I've seen. And again, there can also be highly custom systems, which may be more than that, depending on the functionality. So it definitely can range. I would just say, again, kind of going back to not all IRT platforms are the same. Just make sure you find a platform that meets your project's requirements is critical.
Right, right. We do still have quite a few questions. I'm trying to look through and pick maybe just a couple more. And again, whatever we don't get to, we'll try to follow up via email. So if you have additional questions, go ahead and submit them. This question looks like it's for Susan. Are some randomization schedules more common in certain therapeutic areas? And then they're giving oncology, for example.
That's a great question. Some randomization schedules can be more common based on the therapeutic area, but there usually isn't a set rule in place. For example, minimization algorithms are often used for more rare disease studies as you're trying to enroll very few subjects spread out across the country or around the world. Ophthalmology is another example, as it has some unique randomization practices. In some particular studies, such as ocular allergy studies, they'll often include both eyes in the study, and one eye will receive active treatment and the other eye will receive placebo. So then they actually have to randomize within each subject. So 100 patients means 200 treatments in those particular cases. So it can get quite complicated, and there's often error because patients will switch and dose the wrong eye with the right product.
Oh no.
That's never good.
Never good. And speaking of that misdispensation that you were just talking about, and you both mentioned it during the presentation, somebody asked, how often does misrandomization or misdispensation happen?
I think that's a question both Jen and I can respond to.
Yeah.
So for me, I've seen it fairly rarely in my career, but it does happen. And again, then you have that flurry of activity to make decisions on how you're going to manage that from an analytical perspective. Jen, what are your thoughts?
Yeah, I would agree with Susan. It's very rare. I think the benefits with an IRT is you're notified in real time. So when Susan talks about that flurry of activity, it may be more of a choreographed flurry of activity with an IRT, and an IRT can, especially as we talked about, an IRT plan can kind of minimize that risk of the impact of future subjects and minimize unblinding because you have a plan in place for addressing it when and if it occurs.
Great. Well, thank you so much. Thank you, Jen and Susan, and thank you to all of our webinar participants who are logged in. I will leave the webinar portal open for a few more minutes to round out the hour. If anybody has additional questions, go ahead and submit them, and we'll follow up with everyone we weren't able to get with via email. So thank you again for joining us for today's webinar. I hope you enjoyed it. I hope you learned about patient randomization and the role of IRT in clinical trials. And we will follow up via email with the recording so you can all watch it again if you would like, or share it with your colleagues. Thank you again. Have a great day.
Thanks, everyone.
Thank you.